Good morning, my dear students. Welcome to our zoology online class. Dear students, in the last class we started the fourth chapter, and our chapter name is Principle of Inheritance and Variation. So we have started the fourth chapter in the last class, and we have completed two important topics also: multiple alleles and RH factor. Okay, multiple allele and RH factor. So these two topics we have completed in the last class. So today we are going to study about some more topic. Okay, so first we are going to study about one disease. Okay, the name of the disease is called erythroblastoma fetalis. Erythroblastoma fetalis. Listen very carefully. It is very important for 5-mark question. Here, erythroblastoma fetalis meaning it is a disease. So the reason for this disease is RH factor. Okay, in the last class we studied about the RH factor. So RH factor playing a very important role during childbirth or during the pregnancy. Okay, so RH factor playing a significant role during childbirth. Listen very carefully. If a mother is in RH negative condition and the father is in RH positive condition, the developing fetus may be in RH positive condition. Okay, fetus meaning you all know the baby developing inside the uterus of the mother is called a fetus. Okay. So, if the mother is in RH negative condition and the father is in RH positive condition, the fetus may be in RH positive condition. Okay, so when the RH negative mother carry a RH positive fetus in her body, sometimes the immune system of the mother is sensitized. Okay, sometimes the immune system of the mother is sensitized and producing one antibody. Name of the antibody is called IgG antibody. Name of the antibody is called IgG antibody. Okay, so this antibody passing through the placenta and it is finally mixed. A small amount of this antibody is crossing through the placenta and mixed with the fetus blood of the fetus. Hope you understood this point. So the mother is in RH negative condition, father is RH positive. So the developing baby also may be in RH positive condition. So when a RH negative mother carrying the RH positive fetus, the immune system of the mother is sensitized and producing IgG antibody. Okay, and this a small amount of this antibody is crossing through the placenta and mixed with the blood of the fetus. Okay, so it's happening in the first delivery or first delivery time, and it will not cause any problem to the fetus during the first delivery. Understand you? Hope you all understood this point. So during the first delivery time or for the first baby, it will not cause any problem. But the subsequent pregnancies with the same mother, RH negative mother, attend pregnancy for the second time, third time, fourth time continuously. Okay. So the following pregnancies, when the same mother, RH negative mother, carry the RH positive fetus, some antibodies, anti-D antibodies are produced in her blood, and these antibodies enter in the blood of the or mix with the blood of the fetus. So the next baby, second, third and fourth babies may be affected by some diseases like jaundice and anemia. Okay, here anemia meaning is a disease affecting the RBCs. The RBCs is breakdown. If any babies or anyone is affected with this anemia disease, the RBCs are highly damaged. Okay, so here the subsequent babies affected by some diseases like anemia and jaundice. Understand you? So this condition is called erythroblastoma fetalis. This condition is called erythroblastoma fetalis. So hope you have understood this point. So for the first delivery, for the first baby, it will not cause any problem. But for the second, third, fourth babies, it leads to some disease. Okay, because antibodies crossing through the placenta and mixed with the babies. Okay, baby's blood. So it leads to some diseases like anemia and jaundice. This condition is called erythroblastoma fetalis. Okay, prevention of this erythroblastoma fetalis. We can prevent this erythroblastoma fetalis. Okay, so to prevent this erythroblastoma fetalis disease, anti-D antibodies should be injected in the body of the mother during the 28th and 34th week of gestation. Okay, so totally 40 weeks are the pregnancy period or gestation period. Okay, so to prevent or to avoid this erythroblastoma pyogenes disease, anti-D antibodies should be injected in the body of the mother during the 28th and 34th week of gestation. Understand of you? Understand? And whenever she attained pregnancy, soon after the delivery, this antibody should be injected. Okay, so the antibodies should be injected during the 28th and 34th of gestation, and it also should be injected soon after the delivery. Okay, we have to do this injection. We have to inject these antibodies whenever she attains the pregnancies. Understand? So when we inject this anti-D antibody, passive immunity power is developed in her body. Okay, so it will prevent the production of the antibodies. Understand of you? Understand? So to prevent the erythroblastoma pyogenes disorder, anti-D antibodies should be injected in the blood of the mother. Understand of you? Understand? So dear students, now we have completed the first topic, erythroblastoma pyogenesis. Hope you are understood. Then the second topic is sex determination. The second topic is sex determination. See, you all know the chromosomes will be determining the sex in maximum living organism. Okay, so genes are present inside the chromosomes. Okay, some genes are controlling the sex. Understand of you? So these genes are located inside the chromosome, and that chromosomes only determining the sex in maximum organism, and these chromosomes are called allosomes. These chromosomes are called allosomes. Understand of you? Actually, the sex chromosomes are classified into two types. Okay, so the similar gametes, in some organisms, if the male produce similar sex chromosome, the female producing dissimilar sex chromosomes. If the male producing dissimilar chromosomes, the female producing similar chromosomes. Understand of you? So hope you understand. So in some organism, the male is producing same chromosome. In the female body, dissimilar chromosomes are there, or in the male body, dissimilar chromosomes are there. In female body, similar chromosomes are there. Okay, if the both chromosomes, sex chromosomes are similar, it is called homogametic, and if the chromosomes are in dissimilar condition, it is called heterogametic. Okay, this condition is called heterogametic. Hope you understood this point. So listen very carefully. See, the first we are going to study about heterogametic male. Hetero means already I told you difference. Okay, both sex chromosomes are different. So here heterogametic male means in male organism both sex chromosomes are different. Both sex chromosomes are different. There are two examples I have written here. The first example is XX and XO type of sex determination. XX and XO type of sex determination, and we can see this type of sex determination in cockroach. So example for this XX XO type of sex determination is cockroach. Hope you understood this point. Then, so listen very carefully. See, in this diagram I got drawn the sex determination. See, female. Okay. So here AA meaning autosomes. So what are the chromosomes determining the character, body character or somatic character? That chromosomes are called autosomes. Okay, and what are the chromosomes determining the sex? That chromosomes are called allochromosomes or allosomes. Okay, so here AA means autosomes. So in female, both chromosomes are similar. Both chromosomes are similar. XX. Okay, but in male, one X chromosome and one O chromosome. Okay, so here in female, the both chromosomes are X chromosomes. So female producing only one type of sex chromosome. Both one type means X chromosomes. Understand a few? But here in male, one X chromosome and one O chromosome. So listen very carefully. Here these two are the gametes. One autosomes and X chromosome. Here one autosome and one X chromosome. Here one autosome, one X chromosome, and one here autosome and O chromosome. Okay, so when a gamete carrying this X chromosome, okay, the female gamete carrying the X chromosome fuse with the gamete carrying the X chromosome from the male, so it has two X chromosomes. Okay, so here the young one is or progeny is female. Young one or the progeny is female. Here from the female, one X chromosome is coming, and from the male one O chromosome is coming. So here XO, so that young one is male. Young one is male. So in this type of XX, XO type of sex determination, the female producing single type of sex chromosome X, okay, but the male producing two types of X chromosome, one is X and O, okay. So the male producing two gametes, okay, two gametes. One gamete consists of X chromosome and the second gamete consists of no X chromosome, and in the second gamete there is no X chromosome. But in female producing two gamete, okay, female also producing two gamete, but the both gamete consist of X chromosomes. Understand of you? So hope you understood this XX, XO type of chromosomes, type of sex determination. And the second example is XX, XY type of sex determination. Example Drosophila. Example Drosophila. So in this type of, this is also one of the example for the heterogametic pair. So females are producing similar gamete and the male producing dissimilar gamete. So here XX is the female gamete. So female producing two gamete, only one type of gamete but two gametes. Okay. So both gametes are consist of X chromosome. But in male producing two types of gamete. One gamete consists of X chromosome and the next gamete consists of Y chromosomes. Okay, next gamete consists of Y chromosomes. So here from female one X chromosome is coming and from the male also one X chromosome is coming. So both chromosomes are X chromosome. So it is a female. Suppose from the female one X chromosome is coming and from the male one Y chromosome is coming. So the young one is getting one X chromosome and one Y chromosome. So the young one is male. So the young one newly produced young one is male. So these two are the example for the heterogametic male. Hope you understood this one. Then next one is the heterogametic female. Heterogametic female. So hetero means difference, already I told you. So the male producing similar gamete and the female producing dissimilar gamete. Okay. So listen, here two examples I have written here. One is XO, ZO, Z type and ZW, Z type. So listen carefully. So here in female two autosomes are there and one Z chromosome and one O chromosome. In male both chromosomes are Z chromosomes. So here female producing two X or two gamete. One gamete consists of Z chromosome and the second gamete has no Z chromosome. Listen very carefully. The female producing two gamete, two eggs. Okay, the first eggs consist of Z chromosome and the second egg there is no Z chromosome. Here the male producing two sperm. The first sperm consists of one Z chromosome and the second sperm also consists of one Z chromosomes. Understand of you? Understand? So when we draw this phenotypic character, so suppose if we get two Z in the the progeny of the young organism getting two Z gamete, two Z chromosome, then it may be male. Understand? And suppose the young ones get one Z and one zero chromosome, the young one is female. The young one is female. Understand of you? Understand? And the second example is ZW, Z type of sex determination. Example, bats. In bats we can see this type of sex determination. Okay. So here the females getting one Z chromosome. Female producing two eggs. Okay, two eggs. Understand of you? So here female producing two eggs. One egg consists of Z chromosome and the next egg consists of W chromosome. But the male producing two sperms, both the sperm consist of Z chromosome. Both sperm consist of Z chromosomes. Hope you understood here. That here, say, suppose the newly produced young ones gets one Z from the mother and one Z from the father. So both are Z chromosome. Okay, both gametes consist of Z chromosomes. Understand? So it is a male. It is a male. Then suppose the young ones getting one Z chromosome from the mother and one W chromosome from the father. It will get one Z and one W chromosome. So the young one may be female. The young one may be female. Understand of you? So these two are the example for the heterogametic female. So for heterogametic males means male has two dissimilar gametes and the female has similar gamete. Here heterogametic female meaning the female have two dissimilar gametes and the male have similar gamete. Understand of you? So hope you are understood this type of sex determination. Okay, then now we are going to study about sex determination in human being. We are the example for the heterogametic sex determination. Okay, because you all know in female body 2 X chromosomes are there, and in male body 1 X chromosome and 1 Y chromosome is there. Actually, our body cells consist of 46 chromosomes. 46 chromosomes. All these 46 chromosomes are arranged in 23 pairs. All the 46 chromosomes are arranged in 23 pairs. Here the first 22 pairs of chromosomes determining our body character, so they are called autosomes. And the last pair, okay, the 23rd pair of chromosomes determining our sex, so it is called allosome. Okay, so 46 chromosomes are there and it is arranged in 23 pairs. First 44, that means first 22 pair determining our somatic character, and the last pair, 23rd pair, that means the 45th and 46th chromosome determining our sex. Understand of you? Understand? So listen very carefully, my dear students. So this is the father's sex chromosome. Here 44 autochromosome and 45th chromosome is the X chromosome and 46th chromosome is the Y chromosome. And in mother 44 autosomes are there and 45th chromosome is the X chromosome and 46th chromosome is also X chromosome. So it is arranged, and I told you all the chromosomes are arranged in 23 pairs. Okay, so here we can get 2 gametes. 22 autochromosome and 1 X and 22 autochromosome and 1 Y chromosome. Then in mother, in female, 22 autochromosome plus 1 X chromosome, 22 autochromosome plus another 1 X chromosome. So when we draw the genotype, we will get 44. Suppose if we get 44 autosome plus 2 X chromosome, the baby is a female. Suppose if we get 44 autochromosome and 1 X chromosome and 1 Y chromosome, the baby is male baby. Okay. So suppose this, just imagine it's a female baby. Okay. So female baby getting 1 X chromosome from the father, and from the mother also the baby is getting X chromosome. So 2 X chromosomes are here, so the baby is female. And here the baby getting 1 Y chromosome from the father and 1 X chromosome from the mother. So 1 X chromosome and 1 Y chromosome, so the baby is male baby. So this is the sex determination in human being. Understand, my dear students? So hope you are understood the sex determination. So now we have completed 2 topics. The first topic is heteroplastic fetalysis and the second topic is sex determination. And the next, the third topic is war body. The next topic is war body. See, in the year of 1949, 2 scientists, one scientist name is Bar and the next scientist name is Bertrand. So these 2 people discovered a thick or condensed body or structure in the nerve cell of the female cat, and it is completely absent in the male cat. Okay, so in the year of 1949, Bar and Bertrand, these 2 people discovered a thick or condensed structure in the nerve cells of the female cat, and it is completely absent in the male cat, and they called this structure sex chromatin. They called this structure as sex chromatin. And later it was called as war body. Later it was called as war body. Understand of you? Understand? So listen here, students, very carefully. See, in XXXY type of sex determination, XXXY type of sex determination, the female possesses 2 sex chromosomes. Female cells consist of 2 X chromosomes, but in male sex cell only 1 X chromosome only there. So male have 1 X chromosome and the female have 2 X chromosome. So here a doubt is arising. One question is arising here. How this male compensates a sex chromosome? Because in female body 2 X chromosomes are there, but in male body only 1 X chromosome only there. Okay. So fine. So this is the question arises further. So finally, after many research and many experiments, they came to a conclusion. And finally, they said in female body 2 X chromosomes are there, but 1 X chromosome is in inactive condition, and the second X chromosome only in active condition. But in male, only one X chromosome is there. That chromosome is in active condition. So in female, one active chromosome is there, X chromosome is there, and in male also one active X chromosome is there. Understand? So this is the conclusion. Understand? Then another scientist, his name is Mary Layard. This person said this barbary is a very strong coiled. Okay, so it's a thick coiled substance. It's a visible substance. Okay, visible substance. And this barbary is completely absent in XO type of females. Okay, XO type of females. And one barbary is present in XXY type of male. Here XXY type male means it's a syndrome. Okay, it's a different type of one problem, one syndrome arising in the male. Okay, so later we will study about this. So in the female having XO type of sex chromosome have no barbary, but XXY type of male having one barbary. Understand? That's clear. So it's the important 3-mark question. Then next the last topic is haplodiploid sex determination. Haplodiploid sex determination. And we can see a special type of sex determination in some insect like honeybees. Okay, in some insect like honeybees. So that type of sex determination is called haplodiploid sex determination. Haplodiploid sex determination. So listen very carefully. So you all know in the previous classes you have studied there are 3 types of honeybees are togetherly living in a honeybee colony. A single queen bee, and more than thousands of worker bees, and more than hundreds of drone bees. So there are 3 types of honeybees are living togetherly in a colony. Understand? Here, these queen bee and worker bees, both bees are female. Both bees are female, and they are developed from the fertilized egg. Okay, and their chromosomes are also in diploid condition. Their chromosomes are in diploid condition. But the males are developed from the unfertilized egg by parthenogenesis process. And they have haploid number of chromosomes. So the female, the queen and the worker bees having diploid number of chromosomes, and the male drone bees having only haploid set of chromosomes. So this type of sex determination is called haplodiploid sex chromosomes. Understand of you? Understand? So the honeybees, the honeybee colony, worker bees and the queen bees are developed from the fertilized egg by reproduction, sexual method of reproduction, and they have diploid number of sex chromosomes, diploid number of chromosomes, and the male bee, the drone bees are developed from the unfertilized egg, and they developed by parthenogenesis process, and they have only haploid set of chromosomes. Understand of you? Hope you understood this part. Now we are going to study about one important 2-mark question, kin selection. Kin selection, listen very carefully. It's a very important 2-mark question. Here, kin selection meaning in honeybee 2 types of females are there. Okay, I told you. Queen bee and worker bee both are females. Okay, so here the queen bee only laying the egg. Worker bees are female, but they will not lay any egg. Okay, so in a honeybee colony, the queen bee's main function is laying egg. Understand? So the queen bee is laying egg, but the worker bees are not laying any egg. But the worker bee is supporting the queen bee to produce more number of egg for the colony. Understand? So indirectly, the worker bees are supporting to the queen bee to produce or to lay more number of eggs. So this phenomenon is called skin cells. Okay, this phenomenon is called skin cells. Hope you understood. So in a honeybee colony, the one and only function of the queen bee is egg laying. Okay, they are females, so they are laying more number of egg. But the worker bees also females, but they will not lay any egg. They will help or support the queen bee to increase the reproductive capacity or to increase the number of egg to the queen bee. So this phenomenon is called kin selection. Understand, my dear students? So today we have completed some topic. The first topic is erythroblastosis fetalis, and the second topic is sex determination. Third topic is barbody, and the last finally now we have completed haplodiploid sex determination. Understand, my dear student? So this erythroblastosis fetalis is a very important 5-mark question. This sex determination 3-mark question. Okay, heterogametic male, it's a 3-mark question. Heterogametic female, okay, how the sex is determining human beings, like that. Many 3-mark questions are there, 1-mark question also there. The Barbary is a 3-mark question and chemical selection is a 2-mark question. Okay, dear students, today itself try to complete these topics and we will meet in the next class. Thank you.